I'd like to welcome everybody to yet another WestGrid lunch hour seminar presentation. This time it's coming to you from University of Alberta campus, and our speaker for today is Dr. Andriy Kovalenko. And the talk is titled A Closer Look at the HPC behind Multiscale Theory and Modeling. And so Dr. Andriy Kovalenko is Adjunct Professor here at the University of Alberta and also a Senior Research Officer at the National Institute for Nanotechnology. His research focuses on developing fundamental methodology of multiscale theory, modeling, and simulation capable of predicting the behavior of complex nanomaterials and nanosystems, and applying this predictive methodology to provide understanding and rational design of realistic systems and processes of crucial importance in the nanoscience and nanotechnology. In today's talk, Andriy will discuss how WestGrid and Compute Canada resources support and advance his research investigations. And from now, I will carry on.
Good afternoon, everybody. Try. You want to put it in here, okay? Good. So I would like to begin this talk with thanking WestGrid for this opportunity to present what the methodology and applications what we have been doing, and of course for the resources coming from the WestGrid, and for the whole support for these calculations, because HPC is an essential component in what we do, and it's a platform of multi-scale theory and modeling and simulations. This platform couples together the methodologies ranging from deep electronic structure methods, statistical-mechanical theories, and towards continuum solvation theories. And an essential thing that couples all them together is actually statistical mechanics, some theory work, in particular integral equation methods. And this gives us really capable, really powerful tool capable of addressing all those systems that are listed, just areas listed on top of that nice Canadian type landscape, from deep ice to warm greens. And unfortunately, I cannot point at this with the pointer, so I will describe it in words. But when thinking of components of theoretical modeling, I always I have to position ourselves in that landscape from expressions like, oh, theories are the most important component, anything else is not important, to the other extreme, saying somebody of the big names said, never think, always compute. And so where are we on between those extremes? So I always recall something I heard once from Professor Ulrich von Hansmann, who is the developer of multi-canonical Monte Carlo, and he once said, before spending a week of calculations with Monte Carlo, go for 3 hours to the library to just find out the proper way, and then your computations are enhanced orders of magnitude. And then with those high-performance computing resources, you can reach really big results addressing real systems. So we indeed have a coupling between methods and resources. And so the presentation plan is this. I'm going to give you some description of the theories we are using in a nutshell, and then illustrate this with generic models. And important applications of this are in quantum chemistry. The theories coupled with quantum chemistry methods, it's called QC, and major bullet 2. And it applies to nanochemistry in solution something other methods cannot do, except for quantum mechanics / molecular dynamics, but that is becomes hopelessly slow. And then we have coupling with molecular dynamics, and if I have time, I present some nice pictures with applications. So first of all, what is this methodology we are using? It's called molecular theory of solvation. It is based on statistical mechanics. So the top line gives one of the basic equations. It's a Dyson-type equation which gives you the probability of finding particles of liquid around your structure. So on the right-hand illustration, it's a organic rosette nanotube. Such nanostructures are made at the National Institute for Nanotechnology and Department of Chemistry, and solvent distributing around them is presented in this red and white clouds, and this is water. So that equation at the top of the page gives you the probability of the distribution. So the solvation structure, something that would come out of molecular dynamics. However, the molecular dynamics has to sample the whole distributions of these, of molecules and, for example, of some drug molecules in that solution around the nanotube by directly sampling the phase space. So you have to have these molecules, just sample how they move around the nanotube. Whereas this theory does this on paper analytically beforehand. So you go to the library, switch your brain on, and write those equations down, and they give you the distribution function, they give you correlations based on the solvent properties, and they give you the solvation thermodynamics in the closed analytical form. They give you the free energy. So you know how the system evolves based on the free energy, and it's a simple expression which is under just the first formula under bullet 3, solvation free energy, and then you can extract entropy, solvation forces, and partial molar volume. So everything from solving these equations. So instead of instead of directly sampling the phase space, you solve integral equations, and they do this for you all at once. What's the price for that? The price is in the fact that you get an infinite chain of equations, and you have to limit yourself to one, and then to do something which is called decoupling, and then to close your system of equations. And it's being done with some closure relation. Here is my own closure relation, which nicely works for systems of interest. So for details, you can go to textbooks that are available for this area. So a very basic example, simple system. It's sodium chloride, which we use on everyday basis. So sodium and chloride ions. We solve these equations, get the distributions pictured as 3D maps in those four cuts, cross sections, and then based on them we see how the molecules are distributed, water molecules, and how they form bridges between those ions on the lower left cartoons, and they represent all the features of hydrogen bonding in this system. And if we have these two ions in contact, water forms hydrogen bonding around them. If they pull them one from another, we have it's barely seen, but in the lowest cartoon we see a formation of a cavity. So your system works against the pressure, and you have the dissolution effect. And then, if you increase the concentration of this salt, clusters form like those in the cartoon between the graphs. And these clusters have hydrogen bonding between the species. So everything is in there. This is the simplest toy model, generic example. Now we can replace sodium chloride ions with nanotubes and whatever. The methodology will marvelously work on those systems. Now we then thought, why don't we couple this with quantum chemistry? And again, a little bit more formulas. So we have the Kohn-Sham density functional theory, standard thing that are in many packages, but now we couple this with our methodology accounting for solvation effect. And we have the cell consistent field equations. They are in the lower rectangular box. The standard Kohn-Sham density functional theory equations, but plus with an additional solvation term, the last term in the parentheses. That's the contribution of the effect of your solvent on the electronic structure of your molecule in the solvent, which you describe with quantum equations. And you have again this three-dimensional reference interaction site model for the classical structure with the closure, and they are self-consistently coupled with effective potentials, which are calculated in the self-consistent loop. That is, your water affects your molecule, and the molecule in turn affects the water solvation shell around, and you do this self-consistently. Okay, so here is some slide showing how it works for a metal-water interface. And the interface is just drawn on the left hand side, and in the center you see all those shifts of the Fermi level and of the effective potential and the distributions and all the details, which are very close to the results of Carperinello simulations. So you see, we now can address the complex system. This calculation is with Carperinello methods. This is going to last for months, but with this methodology, you can do this on a workstation very fast, like a couple of hours. But still, we need a powerful workstation. So here we resort to the WestGrid for that. And okay, we have put this in a package, which is Amsterdam Density Functional Quantum Chemistry package. So this methodology is in the package. So what it can do now, it's a cell-consistent field calculation for quantum properties of the system of your choice. It can be nanotube or some solute or chemical reaction in solution or some nanocatalysis in solution. It will do this with a better quality than quantum mechanics / molecular dynamics, because you don't get any noise, but you do get all the predictions for this system, all the physics and chemistry. And you can optimize the system because you get analytical gradient, so you can have reactions in solution. And we ran many illustrations. Like here is one illustration. You have some molecule, organic molecule in solution. It fluctuates between Gaussian and trans arrangements, and then the energetics of these fluctuations, the free energy in different solvents, is presented in the table. So we compared our model, which is line number 2, versus other models validated against experiment. And our model, those two, Cosmo and PCM, are interpolative models, semi-empirical models. Ours is first principle. So you give me the solvent, I'll give you the answer what happens with this molecule in that solvent. And I would like again to repeat, this is now implemented in the Amsterdam Density Functional Quantum Chemistry package. Here is another validation for some organic biomolecule. So again, our results are at the level of high level ab initio calculation. Okay, another example of how it's working. It's here it is applied to actually ionic liquid, and we can reproduce all those nice pictures of layering of ionic liquid and distributions of species around those molecules in liquid phase at the level of Carp-Rinello. Again, we do this on a workstation in a simple calculation. If your system is larger, you go to WestGrid. For toy model, workstation is enough. For a larger thing, WestGrid is absolutely necessary. But if you want to run this in Carp-Rinello ab initio molecular dynamics, that would need months. So we have huge acceleration and the possibility of applying this to systems of real systems of real interest. Hello. Okay. So it's how does it scale up? Here is an illustration of this systems that are accessible with this solvation model. For small solutes, which are like 100 atoms, this red line is conducting screening model implemented in the ADF package. So for small solutes, it's pretty decent, but for large solutes, more than 1 to 100 atoms, it just skyrockets. So if you want a large system, it's very difficult. Whereas our scaling of our systems is we are using fast Fourier transform, and so the scaling basically the resources you need are just stay flat while your system is in the box. And here we can handle for very large systems we deal with, like nanotubes. We can handle with that we can handle that with distributed memory machines, or we can handle that with a large shared memory machine if you want really large system, like big nanotubes. So it can be handled with Hungaby machine. But our scaling is just the scaling of fast Fourier transform, which is n log n. Okay, now the memory requirements we have. Here we have to describe the solvation shells around the molecule, and our memory required for typical systems is just circled in this table. So for a simple molecule, you would need a very small memory. For like some oligomeric chain, here is polyethylene oxide chain, you need a little bit more. For zeolite, you need even more, but for prion protein, it approaches 1 gigabyte. If your system is even larger, then you need larger boxes, and this is where we get to larger machines. So right now, these calculations for these typical systems are very nicely handled with the distributed memory machines and, of course, InfiniBand interconnect, because for fast Fourier transform we need fast exchange with the data in fast Fourier transform, and usually it is FFTW, which is handled with stripping of the memory. This is the standard model which is used in FFTW. So this is one of the Fourier transforms we use. Okay, another combination of this is with molecular dynamics simulations. It doesn't want to follow my push. Oh, okay, it's just slow. So, okay, we did something which is molecular dynamics of a biomolecule steered by solvation forces obtained from this theory. So and this is implemented in Amber. So the structure is this. We have in the center, we have a molecular, the molecular dynamics engine of the amber, to which the structures and force fields are fed, and we add this lower box which says solvation, structure, free energy, and forces calculated from our theory, and then interpolated in between for acceleration, and then these forces drive the molecular dynamics of your central molecule. So, for example, this is prion protein in one of our calculations, in one of our tasks we do here at the University of Alberta for prion neurodegenerative diseases and Alzheimer type-related disease. These are proteins which are evaluated in solvent, but solvent is calculated by using that theory. So you can do this faster by then explicit molecular dynamics by, say, 2 orders of magnitude. And here is one example. All the texting gives the details, but the very simple toy model on the right is alanine dipeptide. This is the simplest thing, which nicely represents some important features. You can see it changes the conformation. It does this flip-flop, and the time for that flip-flop is actually about 100 nanoseconds. And this is known from painful molecular dynamics simulations, and this is an experimental fact that it's about 100 nanoseconds. So to handle, even for this simplified system, to handle 100 nanoseconds in solution takes a lot of computational time. And within this methodology of accelerating the things with molecular solvation theory, we can reproduce that flip-flop very nicely. You can see 2 humps on the curves in the graph showing maximal distributions of the molecule in that flip and flop. And we do this right away, much faster, and the acceleration is actually 2 orders of magnitude. So this is a generic system. But now we take all this methodology and apply to this stuff, like organic rosette nanotubes I talked about. So it's a complex supramolecular nanoarchitecture. You take cytosine and guanine, fuse together. It's top left picture, and they have 3 hydrogen bonds on the left and 3 hydrogen bonds on the right. And with these hydrogen bonds, they form a ring, which is called rosette ring, and then those rings get together by pi-pi stacking and form a nanotube with a channel. And the bottom left picture shows the transmission electron microscopy images of these systems, real systems in reality. They form bunches, they can form butterflies and very nice things depending on what you attach, which functionalities you attach. And on the top, those functionalities are crown ethers, but it can be whatever. And you can see the overall structure, but you cannot see what's inside. But with this theory, you can run these calculations and see what's inside, and I call it the mental microscope. So we run the calculations, have those distributions, analyze them, and you see where these molecules sit. It's pictures, cartoons on the left and in the center, in the middle, and they show you where the molecules are, what the hydrogen bonding strength is, and you can see what the attachment strength. And that water, which actually forms a wetting monolayer inside the nanotube, holds it together with such a strength that if you boil them, they stay. This is a supramolecular structure, like meat, for example. It should decompose. It does not. It stays because those molecules hold them together. And with this theory, we know where they sit and why and how. And we apply the theory to this system, but in another solvent. And I'm trying to get to the next slide. Okay, here it is. With other functionalities attached to on the side, which are alkane chains, just simple hydrocarbon chain in this picture. And it doesn't form in water anymore. it forms, it forms, or in methanol, but it forms in hexane, for example. On the right-hand side graph, it shows that the free energy goes down in hexane. And this is the prediction of the theory. This is what they see in experiment. And we can perform this calculation for such complex systems nicely on the WestGrid. So this is how actually the theory marries this nice resources from the WestGrid, and we can run these calculations. Again, there are two modes how we can do this. For smaller molecules, it's like one node or two nodes. For a very large system, you do need a larger memory with InfiniBand interconnect, and big dream calculations, so to speak, are on shared memory machine, which is ungiven. And another, to conclude this, just another nice illustrations related to Western Canada. It's cellulose nanocrystals. So this is the structure of cellulose. So we have fibers, and they have the inner structure. Those inner structures have some substructure. This goes down to kind of chains that are connected in bunches. And on the top right, you see this portion of a subfiber, let's call it, of native cellulose, which has crystalline regions and amorphous regions. So the amorphous regions is the reason why a piece of wood can bend. But the crystalline regions connected with hydrogen bonding are actually stronger than steel. The only problem is that this material dissolves in water, so to speak. So it's strength, but that strength is strange. So people have been trying to do something with that. And there is a plant actually here, built here in Alberta, one of the two in Canada. The other one is in Quebec. They take this cellulose, hydrolyze, get rid of the amorphous regions again, and have cellulose nanocrystals. And now this is a platform. You can put different functional groups on top and make it into drilling mud or paint additive or potentially some reinforcements for biomaterials. So there is a big hope now to make this into something important for Western Canada in particular. So they asking us how it behaves in different solvents. So we just run this optimization first and see it twists. And this is the twisted optimized structure with all that methodology explained with the molecular dynamics coupled with the solvation theory. And then we calculate the interaction between the two individual nanoparticle cellulose in solution. So these are the particles with optimized adjusted surface charge distributions. They are heavily charged on the surface. And then on top of that, there are some functional groups here as sulfonated sulfate groups, which bear extra charge and are designed to have the interaction specific for some solvent. And then we calculate what's the effective interaction between these species in such a configuration. So I'll just go to the next slide, and this is how these results look like. So on the two-dimensional graphs, you can see the effective interaction between two particles in solvent with the distance between them. So you pull them apart, and you see first the strong attraction, then the barrier, and then the second minimum. And this has immediately implications on how they interact in that solution. So you can have water, for example, you can have polar solvent, benzenyl polar, and ionic liquid, and they all behave in a different manner. And from here, you can see how the structure changes when you have this in solution. And one of the tasks of industry is to disperse those nanoparticles, not to have them aggregate and precipitate at once, but to disperse in solvents, for example, in organic solvents. And then it goes, for example, to disperse it in polyurethane mix. And then it goes to extrusion, and voila, you have a plastic with this additive. But it should not all aggregate, it should disperse. So we know how, we give advices how to modify what to put on the surface to have this happen. And again, with this mental microscope, I can see what exactly happens on the surface, where those sulfate groups are, and how the water distributes, and why this dispersion happens or does not happen, depending on the structure. And we can compare this in different solvents, like in water, and in water with different salt concentration, or for sulfated cellulose. And the final funny but exciting application is, you can see this is the effective interaction depending on the torsion angle. So you have two particles, but then one is tilted, and the black line shows that at 10 millimole concentration it has a slight minimum. This minimum causes the formation of so-called chironematic phase. So you have one layer of those cellulose nanoparticles, and another layer on top, and that another layer, the other layer on top is twisted a little bit, and the next layer is twisted yet more, and you have chironematic twist, and this forms a lattice for ordered lattice for light reflection, and people plan to use this to form some security inks for counterfeit paper. So a very nice multi-billion market. You can now handle things that are in high demand, because it appears that, for example, those RFID tags or counterfeit things are important these days. So here we have some predictions how to trigger and optimize this property. So I showed you the theory, and this theory is statistical-mechanical three-dimensional reference direction site model. It's statistical mechanics. That's the part we switch on, where we switch our brains on, go to the library, do some calculation on paper or some analytics on paper, couple this with quantum chemistry, it's Kohn-Sham DFT and other approaches, with some higher ab initio approaches, it's the next bullet, complete active space STF. All this available in the computational packages. And we couple this with molecular dynamics and with some drug design things. I had absolutely no time, so I'm mentioning this now. We have a large suite of drug design applications, and this is implemented in Autodoc, available to those interested through Autodoc. And please ask us if you are interested. But overall, this very nicely runs on WestGrid facilities. It is parallelized, and so I would like to invite people to try these things, because you will see the difference. You will have something that can address real systems, large systems in solution. So you can have this on WestGrid. We are running this on WestGrid. So thank you very much. I think I used my time, all my time.
Well, thank you very much, Andriy. We really appreciate this. At this point, I'd like to invite, if anybody in a remote site have any questions for Andriy, please pick up the mic, and we'll try to answer them from here. If anybody from the audience has any questions, certainly you're available to answer them. All right, we have a question here from U of A. John?
Not yet. Okay. The question is about the use of GPUs for this technology. This has been our plan, but it is definitely possible. We don't have this particularly implemented in this time, but this is available for GPUs because these methodologies use standard things like fast Fourier transform. So it can be readily implemented. We have this in the plans, haven't got to the place yet, unfortunately, but we have this plan. The question is about use the software and the licensing questions. So the implementation in ADF is officially available from the ADF package. So it would be under ADF license. Then the implementation in Amber, the code itself is available under GPL under Amber tools. So please just go there and use it. As usually, everything which is GPL, everything free comes with no guarantee. So that's natural. We are finalizing the licensing question. I mean, it is in Amber, and it will be the coupling with the Amber module, which is called Sander, will be soon available for public use. The implementation in Autodoc is free, but you have to ask us, is it GPL? And the implementation in Molcast is being implemented, so please ask us. And again, for one more mode, which might be of interest for the audience, is to have some collaborations. And then this is totally up to us. If we set up some collaboration, the code becomes available to our collaborators through us. But for ADF and Amber, it goes through their licenses. And in Amber tools, it's available on the code for molecular solvation theory specifically is available free under the GPL license. This is just one more hint. The methodology itself has been included in the molecular operating environment software from the Chemical Computing Group. It's a major Canadian company developing software for biomolecular, biochemical, biomolecular, like drug design applications and biochemistry. This would come through their license, of course, through the MOE license. So here is the landscape. Implementations in the major packages come through their terms, but for the solvation theory itself, we have a GPL through Amber tools. The code is in there. Thank you.
If there's no more questions, we'll conclude this presentation, and we'd like to thank everybody who attended this. At this point, traditionally we turn the mics or the focus for the local site leads and local technical support for WestGrid sites to answer any questions that's been.
Hey, Wally, we have a couple of questions from remote sites actually.
All right, go for it.
Yusas, do you want to go first?
Yep. Can you say anything about the algorithms that you use for optimization?
In the optimization of the structure.
Sure. Or geometry optimization, whatever you mean by that.
Okay, I will explain on the example of applications for nanochemistry in solutions. You have the molecule, which is in some solvent with some species, say ions, counterions, buffer, maybe drugs in solvent. So the optimization of geometry goes through this standard algorithms of the package, which is Amsterdam density functional. And this package changes the geometry based trying to find the free energy minimum. And now this free energy minimum has a component, a major component coming from solution. For that, quantum chemists provide
We've completely lost audio.
Yeah.
Oh.
Yeah, I believe it's the echo cancellation on U of A's side, so if the questioning sites can mute their mics after they ask the question, that would help.
We'll mute right now.
Okay. Should I repeat? Repeat what you were saying. The of solvent of your molecules in solution is being done with the standard tools of the packages involved. For example, Amsterdam density functional, but with solvent term. So
sorry, we lost the audio again, everybody.
All right, I think we're going to have to just move on. SFB, did you want to try to ask one more question, or your thoughts, or just wrap up?
If I could just maybe ask a question, maybe if the speaker could maybe just make sure you hold the mic fairly close to your, up a little bit higher, and make sure that it's catching the audio okay. My question was, I'm a visualization person, and I was interested in if you have any specific challenges with multi-scale, when you have things on multiple scales, if visualization prevents you with any challenges. You had some very interesting and very compelling images in your slides and things like that. I was wondering if you could comment. Hopefully this will work.
This is actually we have recognized that this is a very important question to present our results, and actually we have done for that, just relying on the something with VMD, but that would be actually very useful and very important to use advanced tools to visualize that. And there are tools specific for different packages. ADF has something, Amber has something, but an advanced tool would be definitely of high interest to provide understanding of what this means, because we have the output is a three-dimensional distribution of solvent, and based on that you see where those molecules are, and one peak of that distribution tells you, okay, your say drug molecule is in there, oriented in such a manner, and for that we need and there is one molecule or two molecules in that peak. So we would immediately need not just visualization, but visualization with post-processing, meaning integrals, volume integrals of that, surface integrals, stuff, something like that. So that would be of high interest, and we don't have this developed too much at this moment, but would be interested in advancing this, definitely.
I'm not an expert in chemistry visualization, but I'd be interested to talk to you about that a little offline if you're interested.
Oh yes, please. That would be great.
Okay, great.
Thank you.
I think we're done.
Any more questions? Okay, we're done. Okay, so one more time, I'd like to thank you, Andriy Kovalenko, and everybody who attended, and we are signing off from U of A.
Thank you very much all for attending and for participation in this.
Thank you.
Thank you.
Bye.
Thank you.
All right.